Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
When you think about the grand sweep of human history, you can probably point to, maybe half a dozen inventions?
Right. A handful of moments that didn't just improve life, but fundamentally changed the entire trajectory of our species.
Exactly. We're talking about those genuine turning points, where the very concept of what's possible just gets completely rewritten.
It's true. And the historical precedents are few, but they're monumental. I mean, think about early humans harnessing fire.
Yeah, that gave us control over energy. It changed everything.
Or, the shift to agriculture. Suddenly we can manipulate our own food supply, which, that's what allowed for civilization.
And then you have the printing press, which just blew the doors off the monopoly on knowledge. Or modern antibiotics.
For the first time, a real defense against infectious disease. Each of those moments represents this massive expansion of human control over the world.
But the moment we're in right now, it feels different. It feels like a leap in scale.
It absolutely is. Because with CRISPR gene editing, humanity hasn't just gained another tool. We've been handed the keys to life's source code.
The source code, that's a perfect way to put it.
We now have the ability to rewrite the foundational blueprint of biology DNA itself with a precision that was, it was pure science fiction just a generation ago.
So for the first time, we're moving from just being observers of evolution to becoming active authors of it. It's a staggering concept.
And that is exactly why we need to do this deep dive today. Our mission really is to take you inside that blueprint.
To really understand it.
To understand the mechanism, how it actually works at a molecular level, the medical miracles that are already happening, and maybe most importantly, the profound ethical and societal challenges that come with having this kind of control.
Okay, let's unpack this. Because when you talk about a technology that lets us edit the genome, you might picture some huge secretive project, right?
Of course.
But the actual origin story of CRISPR is, it's far more humble. And frankly, it was kind of accidental.
It's fascinating, isn't it? How often the biggest scientific revolutions are sparked not by some grand search, but by researchers just looking at something mundane.
And having the patience to see what everyone else just dismissed as junk.
Exactly.
Absolutely.
Yeah.
So the year was 1987. You have these Japanese scientists, and they're studying the common bacterium E. coli.
Nothing special.
And they noticed this strange anomaly in its DNA. They saw these highly repetitive segments of code, like a pattern over and over.
And these patterns were separated by unique, non-repeating segments, which they called spacers.
To put it simply, it's like reading a huge book, and in every few pages you find the exact same short sentence repeated.
But wedged in between is this totally random, unique paragraph. It just didn't make any sense at the time.
And so for years, these things were pretty much ignored. People thought they were just evolutionary leftovers.
Right. Junk DNA that didn't appear to code for anything useful.
It took years and a lot of detailed work for these sequences to even get the name we use now. Clustered, regularly interspaced, short palindromic repeats.
Which is a mouthful.
Yeah, which is why we just say CRISPR. It sounds like something you'd find on a dusty old shelf in a microbiology lab.
It really does. And for nearly 2 decades, that's all CRISPR was. It was a biological puzzle, a curiosity. But then the realization hit.
The spacers.
The spacer sequences. They weren't random junk at all. They were fragments of genetic code from viruses.
So the bacteria were actively taking and storing pieces of the DNA of their enemies.
Precisely. The research revealed that CRISPR wasn't junk, but an ancient, incredibly ingenious bacterial immune system.
Wow.
So when a virus attacks a bacterium, the bacterium captures a small piece of the invader's DNA and stores it. It embeds it right there in those spacer sequences.
So it's creating a genetic most wanted list.
That's a perfect analogy. It's a highly specific memory of every pathogen that has ever tried to invade it.
So it's a memory bank, but how does that translate into defense? How does it actually work?
Well, that memory translates into a targeted attack. If that same virus, or even a related one, tries to invade again, the bacterium instantly recognizes the genetic signature.
Because it has the record.
It has the record in its CRISPR memory bank. And upon recognition, it deploys these specialized tools.
To Cas proteins.
Exactly. CRISPR-associated proteins. The stored viral DNA acts as a guide. directing the Cas proteins to home specifically on the invading virus's DNA. The Cas protein makes a precise cut, and that neutralizes the threat.
It's rapid, highly specific cellular defense perfected over billions of years.
Of microbial warfare, yeah.
It's just incredible that our ultimate genetic tool came from watching this tiny, constant battle between bacteria and viruses. But the real shift, the breakthrough that gave us CRISPR as a tool, that didn't happen until 2012.
That's the pivot point.
Yeah.
The work done by Jennifer Doudna and Emmanuel Charpentier, that 2012 paper was just monumental.
What did they figure out?
They realized that this whole elegant bacterial immune system could be, well, hijacked. It could be repurposed.
So instead of only using it to cut viral DNA,
They demonstrated that you could program the system to target and cut any DNA sequence you wanted in any organism.
that move from a biological defense mechanism to a universal programmable editing tool, that was the quantum leap. Why was that so much more revolutionary than what existed before?
Well, the gene editing methods that came before, like zinc finger nucleases or talons, they worked, but they were bespoke engineering projects.
You mean you had to be a specialist?
You needed highly specialized expertise, they were slow to design, and the cost was just enormous. If you wanted to target a new gene, you basically had to redesign the entire protein from scratch.
So only the biggest, wealthiest labs could even attempt it.
Exactly. CRISPR completely leveled that playing field. The ability to just swap out a small piece of guide RNA to target a new sequence, it transformed the
I mean,
process
from artisanal engineering into
a cheap, modular plug-and-play system. If the old methods took months and thousands of dollars, CRISPR meant you could do the same thing for a few $100s in a matter of weeks, or even days.
in almost any standard biology lab in the world.
Anywhere.
The accessibility alone must have just exponentially accelerated the pace of discovery.
Oh, without a doubt. It's the difference between needing a dedicated factory to build one specialized tool versus having a set of IKEA instructions to build 1,000 of them yourself.
So let's move from the history to the mechanics. Because when people hear gene editing, they often imagine some incredibly complex machine. But the genius of CRISPR is really its simplicity.
It's elegant simplicity. The real genius is that the system separates the targeting function from the cutting function. It boils down to two core components working together.
And these are components that bacteria designed for defense, but now there are molecular surgery teams. So what's component one?
Component one is the guide RNA, the gRNA. Think of it as the molecular bloodhound.
I like that.
It's a short strand of genetic material, custom made by the researcher. You program it to match the specific DNA sequence you want to edit. Its only job is to seek out and bind to that exact spot.
So it's the GPS coordinate for the whole operation?
Perfect analogy. It provides the search function, knows exactly where to go, and once the gRNA locates and locks onto the target,
Component two comes in.
component two comes into play. The Cas9 protein. This is the enzyme. This is the molecular scissors.
And it's physically attached to the guide RNA.
It is. So once the guide RNA confirms the address, the Cas9 protein makes a precise, clean cut across both strands of the DNA double helix right at that spot.
Okay, I get the search and the cut, but here's where it gets really interesting for me. The magic isn't really the cut itself. It's what happens after the cut.
Yes.
We're essentially hijacking the cell's own emergency repair system.
That is the essential insight. We're not doing the repair. We use the Cas9 scissors to cause a break, and then we let the cell's own innate biological recovery process do the heavy lifting.
And a double-strand break in DNA is a 5-alarm fire for a cell, right?
It's a catastrophe. So its machinery rushes to fix it immediately. And scientists have learned to manipulate this natural repair process in two fundamental ways, depending on the goal.
Okay, let's start with the simpler goal. Gene disruption. Just turning a gene off.
This is goal number one. The cell's primary way of fixing a break like this is often a pathway called non-homologous end joining, or NHEJ.
And that's the quick and dirty fix.
Think of it as the cell's emergency quick fix. It's fast, but it's slappy. When the cell tries to stitch the two broken ends of the DNA back together, it often makes little errors. It might randomly insert or delete a few base pairs.
And those small errors, those indels, that's actually what the researchers want in this case.
Precisely, because even deleting or adding a few letters usually shifts the gene's reading frame. It garbles the message.
Like changing one letter in a sentence that makes the rest of it complete nonsense.
Exactly. The resulting protein is non-functional. And that's how researchers create a knockout, a permanently disabled gene. It's invaluable for studying what that gene actually does. You turn it off and you see what happens.
Okay, so that's how we break a gene. But what if the goal is to correct a fault or insert something new? That needs a much more sophisticated repair.
That's goal number 2, gene replacement or insertion. And this relies on a different pathway called homology-directed repair, or HDR.
And this is the cell's high-fidelity repair system.
It is. This pathway is much more careful, but it needs a template to work. It needs a piece of DNA that matches the sequence around the cut to guide the repair.
And that's where the scientists step in with their trick.
Exactly. To get a correction, scientists introduce a synthetic DNA template along with the CRISPR components. So we make the cut, and as the cell gets ready to use its careful HDR machinery, it sees this new corrected template that we've provided.
So it uses our template as the blueprint for the repair.
Instead of just patching the break, the cell uses our corrected sequence as a model, and it copies that new perfect genetic information right into the genome.
We've effectively changed the permanent record. It's like surgically removing a typo from a book and then handing the cell's repair crew a new page with the correct version, which they then integrate seamlessly.
And that's why this is a paradigm shift. The precision is just incredible. We're targeting one specific spot among billions of base pairs. The old methods were like using a catapult.
And CRISPR is a drone strike.
It's a laser scalpel. And because the guide RNA is so easy and cheap to make, a researcher can design and test a change to a totally new gene in just a matter of days.
The speed of iteration is just. It's why we've gone from a bacterial immune system to human clinical trials so astonishingly fast.
That's right.
So let's move away from the lab bench and start looking at the human impact. Let's talk about the diseases that are starting to fall to this technology, the applications that really do sound like science fiction becoming reality.
The first and most compelling impact is on those devastating single mutation diseases, the ones caused by a single typo in the genetic code. CRISPR is perfectly suited for this.
And the flagship example has to be sickle cell disease.
It absolutely is. Sickle cell is this terrible lifelong disorder. It's caused by a single point mutation in the gene for hemoglobin.
And that one tiny error changes the shape of red blood cells.
It forces them into this distorted crescent shape.
Yeah.
And that leads to chronic, excruciating pain, organ damage, and a much shorter life.
For decades, the only real cure was a bone marrow transplant, which is incredibly risky and requires a perfect match, so it's not an option for most people.
Right. But CRISPR is changing that entirely. In these groundbreaking clinical trials, doctors are taking a patient's own stem cells.
The ones that make all the blood cells.
Exactly. They take them out of the body, use CRISPR to correct that single sickle cell mutation, and then they return the edited, healthy cells to the patient.
And because you're using the patient's own cells, there's no risk of rejection.
And by fixing the error in the stem cells, you are permanently fixing the entire blood system. The results are, well, they're nothing short of miraculous.
I read one patient described it as being reborn.
That's a common sentiment. These are people who spent their lives in and out of the hospital, and now they're living symptom-free, producing normal red blood cells. Their lives are completely transformed.
And the same approach is working for related conditions like beta thalassemia.
Another serious blood disorder, yes. The genetic fix is very similar, and it's freeing patients from a lifetime of regular blood transfusions.
But beyond just fixing genetic errors, CRISPR is revolutionizing how we fight our most challenging illnesses, and that includes cancer.
Yes, this is moving into the realm of custom-made treatments. We're talking about CRISPR-enhanced immunotherapy.
So how does that work?
It's a really powerful concept. Essentially, we're editing a patient's own immune cells, their T cells, to make them hyper-efficient at hunting down and killing tumors.
Because cancer is so good at hiding from the immune system.
It's brilliant at it. It camouflages itself, or it puts these chemical brakes on the immune system to stop the attack.
So we're using CRISPR to remove the brakes and sharpen the immune system's vision.
Precisely. We can edit the T cells to make them recognize unique markers on the tumors with just unprecedented aggression.
People call them living drugs.
And it's a perfect term, because they don't just provide a one-time fix. They persist in the body for years, providing ongoing surveillance. If a few cancer cells try to come back, these super T cells are waiting to eliminate them.
That idea of a permanent circulating defense against your own cancer, that's a breathtaking shift in medicine. But the research is exploding in other areas too.
Think about diseases in tissues that are hard to reach. For certain forms of genetic blindness, scientists are developing ways to edit genes directly in the eye, since it's a relatively contained and accessible organ.
What about conditions like muscular dystrophy?
There, the research is focused on restoring the production of critical muscle proteins. It's challenging because you have to edit a huge amount of tissue, but the proof of concept studies are very promising. And then there's real hope for neurodegenerative disorders like Huntington's disease.
Which is just devastating. It's caused by a toxic protein from a single mutated gene.
It's an incredibly aggressive disease, and if you inherit the gene, you will develop the disorder. The potential here is to use CRISPR not just to slow it down, but to edit out that harmful gene sequence early, maybe even before symptoms appear.
And what about the effort to cure HIV? How does that fit in?
It uses the same gene disruption concept we discussed. HIV needs a specific receptor called CCR5 to infect our cells. But some people have a natural mutation in their CCR5 gene that makes them highly resistant to HIV.
So the idea is to use CRISPR to mimic that natural resistance.
Exactly. Edit the CCR5 gene in the immune cells of HIV patients, and you essentially lock the door so the virus can't get in. You offer a cure, not just management.
The implication is just staggering. We're moving toward permanent cures for diseases that have plagued humanity for centuries. But this also brings us to that first major ethical flag. The prospect of editing these mutations not in a living patient, but in an embryo before it's even implanted.
And that's the question. Are we just fixing the sick, or are we starting to design the well?
The potential to eliminate suffering is breathtaking, but the applications don't stop with human health. In fact, the agricultural and environmental uses may be just as transformative on a global scale.
This often gets overlooked in the rush to discuss human medicine, but agriculture is where CRISPR is already seeing massive, rapid adoption.
And it's desperately needed. Our food system is under enormous pressure from population growth and climate change.
Right, and CRISPR offers these precise, rapid tools to adapt. Instead of the slow, decades-long process of conventional breeding, you can make a surgical strike.
Can you give us some specific examples of what's already being developed?
Certainly. We're seeing drought-resistant varieties of staple crops like wheat and corn that could allow farming in increasingly dry regions. We're also seeing disease-resistant varieties that eliminate the need for chemical sprays.
And what about benefits for the consumer, especially around food waste?
Absolutely. Researchers have successfully developed tomatoes with an extended shelf life by editing the genes that control ripening. When you consider how much food is wasted globally, that one simple edit has profound consequences.
Not to mention the nutritional side, addressing malnutrition.
Yes, rice is a perfect example. It's the bedrock diet for billions, but it often lacks key micronutrients. CRISPR has been used to engineer rice to produce much higher levels of essential vitamins. That's a scalable public health intervention.
And the livestock industry is also being transformed.
On the animal welfare side, we're seeing pigs engineered to be resistant to devastating diseases that cost farmers billions. And on the climate adaptation side, cattle are being engineered for heat tolerance so they can thrive in warming climates.
I heard about creating hornless cattle to avoid the dehorning process.
Exactly. A simple edit that eliminates a painful, stressful procedure.
And then there's environmental restoration, which sounds almost like ecological triage.
It's a last resort tool for major crises. For example, researchers are modifying corals to withstand warmer, more acidic oceans to try and save our reefs.
And the American chestnut tree.
A classic example. The tree was basically wiped out by a fungal blight. CRISPR offers a precise way to edit in resistance genes and potentially restore this keystone species to our forests.
And then we get to what I find to be the most powerful and maybe terrifying application outside of humans, gene drives.
Gene drives are an extraordinary application of CRISPR. Scientifically elegant, but they carry immense risk.
Explain the technique. How is it different?
Normally, a gene has a 50% chance of being inherited. A gene drive uses CRISPR to cheat that system. It ensures an edited gene is inherited by nearly 100% of offspring.
So it forces the new gene to spread like wildfire through a wild population.
Exactly. It bypasses natural selection to guarantee the rapid adoption of a new trait.
Why would scientists want to do something that dramatic?
The motivation is overwhelmingly ethical in intent. Imagine releasing modified mosquitoes that can't transmit malaria or dengue or Zika.
Malaria alone kills hundreds of thousands of people every year.
So if a gene drive works, within just a few generations, you could eliminate the ability of that mosquito population to transmit those diseases, saving millions of lives.
The benefit is obvious, but the risk, I mean, you're altering an entire species in the wild. What stops it from causing an ecological catastrophe?
That is the core concern. You are fundamentally changing an ecosystem with no way to put the genie back in the bottle.
It seems irreversible.
It is. There's intense debate about control mechanisms like reversal drives, but there's no guarantee. The risk of disrupting food chains or the gene jumping to other species is very real. This requires unprecedented global consensus before any large-scale release.
That irreversible, profound alteration brings us directly to the most challenging part of this revolution. We're on an ethical tightrope now, talking about germline editing and the designer baby dilemma.
This is the definitive ethical boundary for humanity in the 21st century. And we have to be very clear about the distinction between the two types of human genetic modification.
Okay. First, there's somatic cell editing.
Correct. Somatic editing targets non-reproductive cells, lung, liver, blood cells in living patients. The change affects only that individual. The edits are not passed on.
And most ethicists see this as just advanced medicine.
Essentially, yes. It's no different conceptually from a complex surgery. But the second type
is germline editing. And this is where we enter uncharted territory.
Germline editing modifies eggs, sperm, or early embryos. Because the change is made to the reproductive cells, it is inherited by all future descendants.
You're making permanent, irreversible alterations to the human gene pool
that echo through all subsequent generations.
And this wasn't just a theory for long. That boundary was crossed in 2018, the He Jiankui case.
That event sent shockwaves around the world. The Chinese scientists announced he'd used CRISPR to edit the genes of twin girls before they were born.
His stated goal was to make them resistant to HIV.
But the global scientific community's response was almost uniformly one of condemnation. It was a massive ethical breach.
But the horrifying takeaway was not just the violation, it was the confirmation that the technology works. Germline editing is possible now.
And this forces us to confront that slippery slope head on. Where do we draw the line between therapy and enhancement?
You can start with the most defensible cases, right? Preventing a devastating disease like Tay-Sachs.
Exactly. If parents carry the gene for a fatal childhood disease, editing the embryo seems like a clear moral good. You're preventing immense suffering.
But once that door is open, what about just reducing the risk of a disease? Like editing genes that increase the likelihood of Alzheimer's or breast cancer later in life.
And now we're on the slope.
Yeah.
You're editing away genetic variations that might never have caused a problem. You're just changing probabilities.
And from there, it's a short jump to enhancement.
That's where it gets significantly steeper. Traits like intelligence, athletic potential, physical appearance, they all have genetic components. If the ability to enhance these develops, should society permit it?
The argument from some is that it's no different than paying for a better school or a private tutor. You're just giving your child the best possible start.
But the societal risk is profound. You risk creating a biological aristocracy. If genetic enhancements are only available to the wealthy, we are deepening economic divides into irreversible biological ones.
You could literally purchase better genes for your children.
That's the fear. A fundamental threat to the idea of human fairness and equality.
And we haven't even touched on the unforeseen consequences of making these permanent edits.
Our knowledge of the genome is, it's vast. But it is far from complete. Genes rarely do just one thing. They have multiple effects.
So a change intended to boost intelligence might unexpectedly impact personality or anxiety or something we can't even predict.
And if we all start selecting for the same set of desirable traits, we run the catastrophic risk of reducing human genetic diversity.
Diversity is biological resilience.
It is. Reducing it could make humanity far more vulnerable to future diseases or environmental changes. We'd be sacrificing long-term survival for some transient ideal of perfection.
And then there are the deeper philosophical questions. Consent. A child can't consent to these irreversible changes to their own identity.
It raises fundamental questions about what it means to be human. Does it change our dignity, our sense of self? These are debates for everyone, not just for scientists.
Despite all this power in the ethical minefield, we need a reality check. The technology is not perfect. Researchers are facing huge practical hurdles.
The hurdles are massive, and the primary focus of safety research is minimizing off-target effects.
So the CRISPR system sometimes cuts in the wrong place.
Exactly. Even with the precision of the guide RNA, the Cas9 can occasionally bind to sequences that are similar but not identical to the target. And those unintended cuts can cause harmful mutations elsewhere.
So you could create a new problem while trying to fix the original one.
That is the paramount safety concern. And to address it, the field is evolving beyond that initial double-strand cut entirely.
This brings us to those newer technologies you mentioned, base editors and prime editors. Why are they safer?
Because they change the whole mechanism. When Cas9 makes that big double-strand cut, it forces the cell into that sloppy emergency repair pathway. That's where many errors come from.
So the trauma of the cut itself is part of the problem.
A big part. Base editors and prime editors avoid that double-strand break. Base editors are like molecular pencils. They can change a single genetic letter, a C to a T, for instance, without severing the DNA backbone.
Correcting a typo without ripping out the page.
Exactly. And prime editors are even more sophisticated. They can directly copy in short, corrected sequences. It's more like direct molecular typing than cutting and patching. They're much safer and much more accurate.
But even if we perfect the editing molecule, there's still the delivery challenge. Getting the CRISPR components into the right cells inside a living person is a huge hurdle.
It is the defining roadblock for most therapies that aren't blood-related. Blood cells are easy. You can edit them outside the body.
But editing the brain or the heart or the liver,
that requires highly specialized delivery methods. The most common are viral vectors.
Modified viruses used as microscopic delivery trucks.
Right. But they can trigger an immune response, and they can only carry a limited payload. The other option is nanoparticle systems, tiny synthetic capsules. They're safer, but we're still working on making them efficient enough.
And specifically for embryo editing, you mentioned mosaicism. Why is that so risky?
Mosaicism is a fundamental challenge. When you use CRISPR on an early embryo, not every single cell might get edited successfully before it starts dividing rapidly.
So you end up with a genetic patchwork.
You get an individual where some cells have the intended edit and others don't, or worse, have a different error.
Which could undermine the whole point or even create new health risks.
Exactly. For germline editing to be safe, you need 100% efficiency in every cell, and we are not technologically there yet.
Finally, let's just address the biological limits. CRISPR is not a magic wand.
It is not. Many traits we might want to modify, like intelligence, personality, athleticism, are not controlled by a single gene. They're highly polygenic.
Meaning they're influenced by dozens, maybe hundreds of genes working together in complex ways.
Precisely. You can't just flip a single genius gene to enter on. Biology is far more complicated than a simple toggle switch.
That context is vital. Let's move to the bigger picture. The social, economic, and political structures are all struggling to keep pace with the science.
The regulatory landscape is just fragmented. And that's a huge problem for a global technology. Different countries have adopted wildly different approaches.
So a procedure that's banned in one country could be legal in another.
And that leads directly to genetic tourism or ethical arbitrage, where controversial procedures move to jurisdictions with less oversight.
So wealthy individuals can just shop for the regulations they like.
That's the direct consequence. In the U.S., for example, federal funding for germline research is banned, but private research might proceed. The EU is much stricter. Other regions are more permissive. It's a dangerous patchwork.
And international bodies like the WHO can only advise. They have no enforcement power.
Right. Creating binding international agreements on something this fundamental is incredibly difficult. The governance systems we have are just not built for the speed of CRISPR.
And that failure is compounded by the most pressing issue of all, cost and access. What is the price tag for that sickle cell cure?
This is the looming tragedy. Those single-dose curative therapies are priced in the range of $2 to $3 million per patient.
Millions of dollars.
And even with future efficiencies, they're likely to remain prohibitively expensive for most of the world's population for a very long time.
How can that be? If public funding supported the basic research.
That's the flashpoint. The fundamental research may be public, but the intellectual property and the immense cost of clinical trials are borne by private biotech companies. They argue they have to recoup those costs.
So these life-saving cures risk becoming available only to the extremely wealthy.
We risk creating a medical system where only the affluent can afford to eliminate generational diseases. And if we ever get to enhancement, that risk is amplified. Genes literally become a commodity.
This isn't hypothetical. We see it with expensive cancer drugs already.
We do. We need deliberate, aggressive steps to ensure broad access. Things like mandating affordable pricing for publicly funded research, or international initiatives like we had for HIV medication.
Though it's worth noting, the upfront cost, while huge, might offer better long-term value.
That's the key economic argument. Curing a lifelong disease is ultimately cheaper than decades of chronic treatment. But accessing that massive upfront capital is the problem.
The same tension exists in agriculture, right? patented crops could widen the gap between industrial farms and subsistence farmers?
The irony is the technology is cheap to use, but the product is expensive to buy. If we can overcome these hurdles, CRISPR could be one of our most powerful tools for global food security.
We've covered so much ground today, from an ancient bacterial defense to the prospect of conscious evolution. And the technology is still evolving at a breathtaking pace.
It's relentless.
Yeah.
Every few months, there's a new, more precise version. We've talked about prime editing, which is much safer.
And now there are systems like CRISPR-Cas13 that go beyond DNA entirely.
Right. Cas13 targets RNA, which carries temporary instructions. This allows for temporary, reversible gene modifications that don't permanently alter the genome. That's a huge benefit for certain treatments.
So what does this all mean for us as we watch this revolution unfold?
What's fascinating is that CRISPR represents a fundamental inflection point. For billions of years, evolution was driven by blind mutation and natural selection. We now possess the ability to read and rewrite that code with conscious intention.
And that power carries a profound responsibility.
It does. The decisions we make now will echo for generations. Getting it right means eliminating enormous suffering. Getting it wrong risks exacerbating inequality or causing irreversible changes we can't undo.
We have to remember, CRISPR is just a tool. It's not a moral compass. Like a hammer, you can build a hospital, or you can tear down a wall.
That is the essential wisdom. Scientific rigor must be guided by ethical wisdom. We need robust, transparent public dialogue, and we need international cooperation to ensure these benefits are shared by everyone, not just a privileged few.
The CRISPR revolution isn't something that's just happening to us. It's a story we are actively writing through our collective decisions. The code of life is being rewritten, and we are all, in some sense, the authors. The question we leave you with is simple. Knowing the power and the potential, what story shall we tell?